Hello, everyone. This is Keshav Sharma. I would like to welcome everyone to my presentation. Today, I will be talking on the environmental change, modulates, the resistance in Triticum aestivum, and its interaction with Puccinia graminis, f.sp. tritici. Going through the quick introduction, wheat is one of the major crop grown around the world. Stem rust is a significant disease in wheat caused by fungal pathogen Puccinia graminis, f.sp. tritici. Yield loss varies usually 40 to 70% in total loss. Breeding for resistance and molecular research are in progress, but emergence of resistance is a major problem. Deployment of varieties with broader resistance is necessary to combat this pathogen. Going through quick pathogen virulence and disease resistance. Outbreak of rust such as UG99 is due to change in virulence by PGT to the widely deployed genes. Various environmental factors such as temperature can modulate the expression of host genes and can alter the resistance. Temperature-sensitive genes such as SR6, SR13, and SR21 induces varying level of resistance with changes in temperature. Temperature does not always change the virulence, but changes the level of host tolerance, leading to varying level of resistance. So quick hypothesis is that is the expression of SR genes altered by temperature? And are they only solely responsible for the host-induced resistance? Also, the temperature does not always change the virulence, but changes the level of host tolerance, leading to the varying level of resistance. And are there similar genes expressed under high or low temperature induced under resistance conditions? Going through quick materials and methods, wheat seedlings, the left picture with as you see here in A is the 1 week old wheat seedlings, and those are inoculated with PGT rays, MCC-FC, and they are induced to the mist chamber. As you see here, the mist coming out from those pipes, and it is kept for 12 hours. After that, these plants are moved to the various incubators where we have the temperature with high and hot conditions would be 25 degrees Celsius at day and 22 degrees Celsius at night. For low and cold condition, 18 degrees Celsius would be for day and 15 degrees Celsius for night temperature. We collected samples for the RNA-seq experiments. Leaf tissue were collected at day 1 and day 3 after inoculation for RNA-seq and qPCR experiments. RNA-seq data was quantified using Callisto, and differential gene expression analysis was performed in RStudio with various bioconductor packages. Pathway analysis and biological function of the differentially expressed genes were conducted via gene ontology, Agrigo-V2 and Shinigo. The infection types were observed and recorded after 2 weeks of the inoculation. So we have some phenotypes here for SR genes. As you see in A, so which is for the LMPG at high and low temperature, for SR6, high and low temperature, SR13, high and low, and for SR21, high and low. We can see here, so SR6 has the drastic phenotypic change with the temperature. It is susceptible at high temperature, but resistant at low temperature. LMPG is susceptible at both conditions. SR13 is has higher resistance at high temperature, but low resistance at low temperature. Similarly, for the SR21, it is high resistance at high temperature, but lower resistance at low temperature, although it has less significant difference compared to other ones. So we did some qPCR analysis of the expression of the genes in different lines. SR6 was upregulated at low temperature, but downregulated at high temperature. As you see here, this is for the low temperature, day 1, CR day 1, cold rust-infected day 1, and cold rust-infected day 3. It is both upregulated. But for high temperature, it is downregulated. For SR13, it is upregulated at cold temperature at day 1, but downregulated at day 3. but it is downregulated at high temp at day 1, but it is upregulated at day 3. However, for SR21, this gene is expressed all the time at all conditions, either low or high temperature. We did some RNA-seq analysis and found some differentially expressed genes in all lines. So we can see here, so the number of DEG is higher in SR6 and 21 line at lower temperature. So both of them are inducing resistance, but both of them has higher number of genes compared to SR13. As you see here in the day 3 as well. The number of DEG at high temperature is higher in SR13 line at day 1. So you can see here at day 1 high temp, SR13 has comparatively higher number of genes. And in SR6 has higher number of genes at high temperature at day 3. The number of DEG under resistant conditions is higher in SR6 line at day 1 and day 3. So that means, as you see here, so SR6 has higher number of genes compared to other SR13 or 21 at day 1. And even for the day 3, under resistant conditions, SR6 has a higher number of genes compared to other. And at high temperature, SR21 has higher number of DEG compared to SR13. So that means if you see at high temperature, so you can see here at high temperature, day 1, SR21 definitely has higher number of genes. And if you see here at day 3, it has higher number of genes compared to SR13. So what were the genes expressed under those conditions? So from the enrichment analysis, we have found that various genes associated to cell killing, defense response to fungus, thaumatic-like proteins, biotic stimulus, PR genes, heat shock proteins, and cell wall organizations, and various other protein kinases were upregulated in SR6 lines under low temperature. So we tried to find some common genes expressed under high temperature for SR13 and 21. So they have aquaporin, a lot of water transport, genes associated with water transport, proton and cation transmembrane transport, protein kinases, PR genes, and ferric reduction oxidase, and other DNA binding and transmembrane proteins. We have also tested some genes associated with the resistance mechanisms, such as salicylic and jasminic acid pathway through qPCR. Under resistant responses, most of the PR genes were upregulated and JA genes were downregulated. As you see here, circled by red, these are all the PR genes for SR6, 13, and 21. It is upregulated in most of the conditions. However, JA genes, it is downregulated in most of the conditions. However, PR genes is very consistent with their expression in SR6 at low temperature. In SR13, few of the genes were downregulated at day 3. And SR21, almost all the PR genes were downregulated at day 1, but under high temperature. But two of the genes were downregulated at day 3. So the conclusion here is that environment can modulate the expression of the effectiveness of the SR genes, leading to resistance or susceptible conditions in PGT-infected wheat cultivars. The temperature of SR genes, the expression of the SR genes, SR6, SR13, were highly variable at high or low temperature, leading to various phenotypes, while SR21 was upregulated at both temperature conditions, as you see before in the qPCR figure 2. So the expression pattern of the PR and JA genes indicate that salicylic acid might be upregulated, and jasminic acid pathways might be downregulated during resistance in PGT-infected wheat lines. The study of temperature-sensitive resistant genes is crucial for crucial factor for targeting deployment of geographical locations and environmental conditions. The SR genes might be the molecular factors inducing resistance at high temperature. However, there could be several genes or several cofactors equally responsible for the expression and resistant responses. So the future direction is that study of significant genes and proteins associated with the resistance induced by SR genes in wheat could help identify the molecular clues inducing temperature sensitivity of the genes responsible for resistance or susceptibility. Functional study of pathogenic elicitators that are responsible for altering host resistance could help elucidate the temperature-sensitive resistance mechanisms. Also, the application of molecular cloning and genetic engineering to explore functional properties of candidate resistant genes. With this, I would like to acknowledge my PIs, Sharia Kianian and Matthew Rouse from Cereal Disease Lab, St. Paul location. Also, I would like to thank our close collaborator, Praveen Basgai from the Department of Agronomy and Plant Genetics, St. Paul, University of Minnesota. And I would also like to thank very much to our close collaborators at in Australia. Since they were very nice sharing SR6 gene sequence so that we could do the gene expression analysis. So I would like to thank Evans Laguda and Tim Hewitt from Shire, Australia, and Peng Jiang from the University of Sydney Plant Breeding Institute, Sydney, Australia. So with that, I would like to thank everyone for listening to my presentation, and I would take any questions you may have. And thank you very much.